Hi everyone, so we're going to try something a little differently today. What we're going to try is what is actually called a flipped classroom. What that means is that I give you the presentation, I give you the direct notes, you guys take notes while you're doing this, and we'll get started. So now here's what I'm going to say, per usual. You can either minimize this into a corner while you take notes on your laptop, in the notes section of course, or you can take it in a notebook or anything like that, whatever way is going to work for you and how you take notes. So we'll take notes today, and then what's going to happen is after we are done with all the presentations of the viruses, we are actually going to discuss all of these. So you need to be coming to class understanding the material that I'm presenting right now. Okay, you ready? Let's get started. Okay, so I know some of you are going to have questions throughout this. This is what I want you to do. If you have a question, I want you to go ahead and write it down. And when we come to class and we start doing all these presentation stuff, that's when I'm going to have you guys ask all your questions. So if you have questions now, that's okay. I want you to have questions. However, make sure that we're going to ask them when you're in class, things like that if you're not understanding. So let's get started. So your notes need to be prokaryotic and eukaryotic. That's what we are going to be talking about with the cells. Yes, I am going to be quizzing you on it. Yes, it's going to be on your quiz on November 5th. So the first thing we're going to talk about is prokaryotic. And if you're following along and if you want to look at the notes after we do this, because there is going to be some portions of links that I want you to click on and things like that, you're going to skip to number slide 88 for me. And that's going to go ahead. I think it's either going to be 88 or 89. You should go ahead and make sure you are in cells and you're in the Excel one, okay? So let's go ahead. We'll get started with prokaryotics and go from there. So prokaryotics are simple cells. That's really what you need to understand about prokaryotics, is that they're simple and they actually have no membrane-bound organelles. And organelles are pretty much these tiny organs in a cell that help make it function, help homeostasis into the cell, that special word, that homeostasis that we're coming back to. These are living things. They're in a cell. They reproduce, things like that. We're going to talk about a couple of different examples of prokaryotics. And we've already actually talked about an example of prokaryotics, even though I haven't specified that. But our prokaryotic that we're going to be talking about, and we've already talked about, is actually bacteria. Now, there are two kingdoms of prokaryotic. What I mean by kingdoms is that there's like two sections, and there's two ways of categorizing these certain types of bacteria. So we have kingdom Eukaryotes, it's real fun, and Archaeobacteria. These are two different kingdoms. Depending on the kingdom is the certain categories, characteristics, structure, things like that that go along with that. And that's why we have two different sections or two different kingdoms, is because they differ in those status. Okay. Again, also feel free to pause this at any time so that you can take these notes. I'm going to continue going on into the next one, but definitely feel free to pause it whenever you need to. I'm just going to keep on going like if I was talking and reading the slides. Okay, so moving on with prokaryotic. Many scientists actually believe that this is the first organism on Earth. And like I said before, these are obviously microscopic. They're unicellular, which you need to know what unicellular means because I'm going to be talking about it so many different times. But unicellular, all it means is that it's one cell. So these are all one cell organisms, and that's our prokaryotic. Now, they're also the most numerous or most abundant. We see them. They're everywhere. And I'm not even saying that to sound as creepy as the viruses. There's a lot of bacteria, things like that. These are actually, a lot of bacteria are good bacteria. Remember that there's only a very small percentage of bacteria that are actually going to be pathogenic, like the E. coli and Streptococcus. There are so many types of prokaryotics and that we do not even know about yet still, and that we're still finding out every single day. So I mean, there's even prokaryotics that are actually in the stomach of termites, which is kind of crazy to think about, but if you think about it, we also have bacteria in our stomach, so it kind of makes sense a little bit. They're found everywhere, and they're actually able to survive extreme environments. Now when I say extreme environments, I mean extreme temperatures, extreme saline content, extreme altitudes, things like that we would never even think that something would survive there. Yet here they are. And that's what makes them absolutely fascinating. So these are the types of prokaryotes that we're going to talk about. That Eubacteria that we talked about in the very beginning, that's our first kingdom. Our second kingdom is going to be Archaeobacteria. And we're going to go into depth with all of these because, again, like I said, that since they are in two different kingdoms, that means that they have to have two different structures and categories and things like that. So we're going to go into a little bit more depth. There's a couple pictures here just to reference something. You have no idea what that's referencing yet, but we're going to get into that. But we are first going to talk about Eubacteria. All right, Eubacteria. Most studied organism. Pretty much there's so many different scientists that look at them and figure them out and trying to figure everything out about them. They're found everywhere except extreme environments, and that's where Archaeobacteria come into. So you can see already the difference between our two kingdoms. Archaeobacteria found in non-extreme situations, Archaeobacteria found in extreme situations. They also have extremely strong walls called peptidoglycan. I never say this right, I'm not gonna lie. However, what this does mean is that they are a mix of sugars and amino acids. Now you should remember, when we are talking about macromolecules, amino acids are the basic unit for, you should be able to answer this before I say it, proteins. Then sugars are going to be the monosaccharides, which are going to be the monomer of carbohydrates. Make sure you understand that. So that's what peptidoglycan, that's cell wall, that's what it treats. So the cell wall is around this bacteria, and it has a specific name, and it's made out of carbohydrates and protein. That's what gives it that really strong wall. Okay, we're done with Eubacteria. We're going to be talking about Archaeobacteria, because there are actually 3 subcategories with our Archaeobacteria. Okay. Here's where there's going to be a little bit more stuff that you need to do that is actually connected with the slide. So if you don't have the slide open, you're probably going to want to open it sooner than later. Now, heliophils. These are usually aerobic, which means that they actually have to have oxygen. And so aerobic, they have to have air, oxygen. Just a little fun little thing. These are actually adapted to live in very salty environments, which is, again, Eubacteria can't live in those salty things. However, Halophilias, which is a type of Archaeobacteria, can. Okay. Now, methogens. These are anaerobes. This means they do not need oxygen to live, so they don't need any air. However, they do need CO2. Now, if you've ever heard of methane, that usually means CO2. This is why we get methane. Methogens, actually, they live off CO2, and then they, as an excretion, when they're eating something, they excrete methane into the air. So you'll find them in volcanoes, swamps, sewage treatments. They're found a lot in any type of plants that are used for that, and then GI tracts of animals, including us. So you know that fart that you just made that smelled really, really bad? That's probably actually caused by those methogens. Yay, now you have something else to blame besides your own self. The next thing is going to be thermoacidophils. These are very strict anaerobes, which means that they die in oxygen. The moment they touch oxygen, they die. So they live in extremely hot and acidic environments, so hot springs and thermal vents. Okay, this is where I would like you to pause this. And you're going to actually click on each one of these. Each one of these has a link. Halophils and thermoacidophils both have a video that you're going to watch. Methogens is actually going to be an article. Now, if you go to the next slide, this is what you're going to be taking notes on with these specific things. Now, halophils, you're going to watch this video. It's called Weird Places, Australia's bright pink lake. And you're going to give me 3 facts about halophils, especially the location, talking about that. In methogens, it's going to open up this nice little article right here, Methane from Microbes, a Fuel for the Future. You're going to go ahead, read the article. You're going to tell me where can these bacteria be found, and then you're going to tell me how can this help with clean energy. What I mean by clean energy is renewable resources. If you have questions about that, come see me. Then you're going to click on thermoacid fields. This is going to give you this wonderful video about Yellowstone, actually. And you're going to answer me 2 questions. What are the color bands? And then what specific color is the thermophilus? If you want to write any other information, you can. However, these are something that we're going to specifically talk about in class. So pause this video, go to those links, answer these questions in your notes. Perfect. See you in a little bit. Okay, so I'm assuming you're back. I'm assuming that you did all that good stuff, and you're going to come to here. Now we are talking about structure of prokaryotes. Now, they're microscopic, they're unicellular. I shouldn't have to go into detail of what those mean. They do have some characters that all cells have. So they have DNA, they have ribosomes, but they do not have a nuclear membrane surrounding their DNA. What I mean by that is they don't have a nucleus. And I know we haven't talked too much about nucleuses. All you need to know is that DNA is just kind of like in there. It's not necessarily bound to a certain place. It's just kind of floating around. They also do not have membrane-bound organelles. What I mean by that is they don't have organelles that necessarily have this. They have things that have purposes, but they're not bound by anything. So again, they're kind of a little more free-floating. Very small. But they're able to carry out all life functions. So they're able to have homeostasis. They're able to get energy. They're able to get to excrete and things like that, and able to actually reproduce, like we've talked about before when we talked about bacteria a couple days ago. Continuing on with that, so they have chromosomes. So their DNA is in chromosome structure. However, they're not bound by anything. So they're arranged differently. They're large circular chromosomes, and they also contain that smaller DNA pieces called plasmids. And this is what we talked about before in our notes with bacteria and viruses. Okay, we're continuing. Prokaryotics, they have a lot of structure to them, obviously. So the next one's capsule. They have they secrete a layer of these polysaccharides, which you should know that polysaccharides are which molecule? Carbohydrates. Know that. Around the cell wall, this is going to form that nice capsule. This keeps the cell from drying out. It helps attach itself to things. It also protects the cell from antibiotics and being killed by white blood cells. Now, this can be a good thing or a bad thing. This can be a good thing if your bacteria is actually good for you and your body is actually thinking it's something that shouldn't be there. Or 2, if it's a bad bacteria that's in your system like E. coli or Streptococcus, they do have a cell that, this capsule protects it. So this is why your body has to work even harder and why you have to introduce antibiotics. Billy! These are cool. These are microscopic hair-like structures that are made of protein. They help attach it or bridging between cells. And then they also are part of this, it's called conjugation. This is where it's sending plasmids to change its genetic characteristics. So this is where it actually kind of mutates, and this is why, because of this, and this gives it its antibiotic resistance. Okay, got a couple more. They're pretty quick though. So now we're going to talk about eukaryotic cells. So eukaryotic cells, we're going to talk a lot about in the next couple weeks, but know this, they're larger, they're more complex. Moving on. They also have a true nucleus, so that holds all its DNA. So it's not just free-floating like it was in the prokaryotics. It actually has it in this nice little ball bundle. This is, where, again, prokaryotic cells don't have that nice nucleus. And then, so prokaryotics, they have what we call the nucleoid, which is just where the DNA is located. Just a location. It's not really anything. It doesn't have like a bound to that area. Okay, keep going. Prokaryotic cells versus eukaryotic cells. DNA in eukaryotic is linear, prokaryotic, circular. Ribosomes. So both cells have ribosomes. Awesome. They both have cytoplasm, and they both have cell membrane. Cell membrane is that outer layer. Please know that. That outer layer, so before the cell wall for most of the bacteria and stuff, now we're talking about this nice little outer layer. Both of them have it. It's cell membrane. It just helps keep everything in, helps keep it not dry out, that stuff. Last one. Here we go. Oh, looks like I think it's the same thing. Oh, look at that. Nicely done. Okay, we are done. Please feel free to watch this as many times over again. Come in with questions. I will see you very soon. If you guys have questions about this, come see me and be kind and be good, and I'll see you soon.